All right, so welcome back, everyone, for this afternoon session. The first speaker today is Matthew Stevens from the University of Chicago. He's going to talk about application of empirical Bayes to the normal means problem. And you're welcome to comment on the accuracy.
Thanks very much. So it's a pleasure to be here. Thanks to the organisers for having me. Let's get started. So this is what I mean by the empirical Bayes normal means problem. I'm going to assume that we have observations, which I'm going to call x or xj, and they're observations, they're noisy observations of some underlying object theta j, and rather amusingly, I'm going to assume that the variances are known. So the Sj there, you can think of those as some measure of the quality of these observations, but I am going to allow them to vary from observation to observation, and that's, I think, quite important for many applications. The variances of different observations, the accuracy of different observations can vary. And the empirical Bayes approach to this problem involves assuming that the thetas come from some underlying distribution G, which we will estimate by maximum likelihood, essentially. And so it's called empirical Bayes because you can think of G here as a prior distribution on the theta values. So it's a distribution from which the theta values are being sampled. And it's called so that's the Bayes part of it, and it's called empirical Bayes because you're estimating G from the data rather than pre-specifying it. And so we'll talk more about estimating G in a minute. The second stage, after having estimated G by maximum likelihood, we can compute the posterior distributions on the theta given G and the observations. And I'm going to assume that the x's given the thetas are all independent and the thetas are independent. So these posterior distributions can be computed one at a time. And the idea is that the idea is that you're essentially borrowing strength or information across observations when you estimate G, and then that comes into that information in the other x's comes in through this G here. So sorry, I'm going to assume that the G comes from some family of distributions, curly G, and there are various different assumptions you might make. So in an introductory class, you might, or even in some applications, you might assume that the thetas come from some normal distribution, or you might go at the other extreme, if you like. You could go entirely non-parametric and say, well, this family is just all real valued distributions, or then there are things that are somewhere in between. And actually, I'm going to focus most of this talk on these last four, or flavors of these last four, which are all inducing some kind of sparsity or shrinkage on the thetas. So I'm thinking about applications where many of the thetas may be at or near zero. So this here is a mixture of a point mass at zero and a normal. So the idea is that if pi 0 were big, then most of the thetas are zero, and then some fraction of them are non-zero, and we assume they're normal. Or instead of normal, you could use a double exponential, or we could go more non-parametric, and consider all scale mixtures of normals or all unimodal distributions centered at zero, for example. So those would be kind of generalizations of these two more parametric ideas. I'll say more about those later, but just some key references. Johnson and Silverman make use of this as well as other prior distributions, and Kunke and Misera do the fully non-parametric case, and I've got a paper where we look at these more non-parametric but shrinkage based. And all these come with software, so the software for fitting these models, and I'll talk a bit more about how it's done in a moment. So yeah, so the focus is on the shrinkage or sparsity inducing priors. So how does, let's assume we've estimated that G. And we're on to the second stage where we're looking at the posterior distribution for theta. I just want to give you the intuition for how the prior distribution G induces shrinkage or sparsity or sparsity in the thetas. So if this is our prior distribution G with a big spike either at 0 or just very near 0. And this is our likelihood here. So we have an observation that's about 1.5 with this kind of standard error here. What's the posterior distribution look like? Well, what happens in this case is that the rising tail of this prior distribution shrinks the distribution towards 0. So this is what I mean by shrinkage. If you have an observation with a larger standard error, you see stronger shrinkage. So the posterior is in green, and it's actually pretty close to being right on top of the prior here, because the likelihood in this case is flat. So that has strong, in this case we would see strong shrinkage towards 0. And here's something in between, where in fact the posterior distribution could be bimodal, because here we've got a prior that's very peaked around 0, and a likelihood that's kind of suggesting the observation may be away from 0. And when we weigh our prior and our likelihood, we get a posterior that has kind of, it's somewhat uncertain whether this observation should be very close to 0 or somewhere away from it. Okay, so that's how the prior induces posterior distributions that are closer to 0 than the likelihood is centered. And so the idea now is that by estimating G from the data, we can get different types of shrinkage depending on what the data tell us. So on the left-hand side, I've plotted the observations and their posterior means in a situation where there's a mixture of sparse signals and big signals. So when we estimate G, we have a G that has a strong mass near 0, but also appreciable mass away from 0. And so what happens is when the observation is close to 0, the posterior mean is very close to 0, and you get this shrinkage towards 0 in this range. But when the observation is far enough away from 0, the posterior determines, as we saw in some of our examples, that this is not one of the 0 entries, and you get much less shrinkage. So you can see the distance from the red line's y equals x. So the distance from the red line is telling you how much shrinkage you're getting. Whereas in this setting, the G is actually just a normal distribution. So there are no 0 effects here. The distribution of effects is just normally distributed. And so you get a different kind of shrinkage behavior here, where everything shrunk a little bit, basically, towards 0. So the idea here is that by estimating G from the data, we can learn how much to shrink and how strongly to shrink. That's and sorry, the axes didn't come out here. But this is a similar plot, but now it's showing how the standard error can affect how much shrinkage is going in. So the previous plots, everything had the same standard error. Now I've got observations with different standard errors. And what you see is the ones with a small standard error, which are more solid red, get shrunk less. They're close to the y equals x line. And then the observations which have a big standard error, which I've colored in a lighter shade of red here, the bigger the standard error, the more it's getting shrunk towards 0. So the idea is that the larger the standard error of an observation, the more the prior tends to dominate over the likelihood, and so the stronger the shrinkage you get towards 0. So there are these two, I think, really nice features of this in terms of a shrinkage-based approach, is that you're estimating how much to shrink from the data with your estimation of G, and you also are able to shrink more for things that have a high standard error than for things that have a low standard error. So if you're used to penalized likelihood type approaches to inducing shrinkage and sparsity, this can be, I think it's kind of interesting that many people don't know about both sides of these, and I myself would say I only know a little bit about penalized based approaches. But if you're used to that way of thinking about it, then you think about it like this: the distribution G is controlling how much to shrink. So it's kind of analogous to estimating G is analogous to estimating the parameters, your penalty, if you like, and what type of penalty should you use in L0, L1, L2. Estimating G is like estimating all this, right? And it's kind of interesting, I think, to note that G is being estimated by maximum likelihood, whereas usually you tune these parameters by some kind of cross-validation. Because okay. So I think that has certain advantages in some ways. You avoid cross-validation, but you have replaced it with a likelihood step. So I said I'd come back to estimating G. So there are kind of traditional parametric approaches. So these have a limited number of, well, in this case, just two parameters, essentially, a prior on 0 and a variance of the normal. So you can do some kind of numerical optimization to optimize the likelihood in this case. But I just want to spend a little time talking about these other approaches which have recently become, I've become interested in. They're kind of less parametric, they have more parameters, but they result in convex optimization problems. So, and they're also more flexible. They include, so for example, the scale mixtures of normals with mean at 0 includes this as a kind of a special case, and in fact you can think of it as the convex relaxation of this family to all mixtures of normals that have mean 0 and different scales. And then an alternative relaxation here is, which includes this one as a special case, is just to say, okay, I'm just going to assume my distribution G is unimodal with a mode at 0. And so it turns out that both these result in convex optimization problems, and the way we implement them is as follows. So basically the idea is, and it's absurdly simple, is to approximate these non-parametric families by a finite mixture of a large number of components. And the more components you use, the better approximation you get, but the longer the calculations take, but they're all convex optimization problems. So, for example, if we just take the scale mixture of normals, we assume G is a mixture of a large number of normals here, where the variances are fixed and known, and they just go from very, very small, let's say 0 can be the smallest if you like, so that's the point mass at 0, and then very, very, very small values up to very, very, very big values. You just take a large number of variances, and then you just have to estimate the mixture proportions. Or, okay, so here's, these are just 3. We can take a larger number, but just to show you how 3 zero-centered normals, give you kind of this kind of shape. Or you can approximate any unimodal distribution by a mixture of uniforms. So you can do the same idea, but assume G is a mixture of a large number of uniforms, all of which either start at 0 or end at 0. So they look something like this. The red ones end at 0, the black ones start at 0. So there's a unimodal distribution. And you can approximate any unimodal distribution arbitrarily accurately by sufficiently large number of uniforms. So the basic idea is to take G to be a mixture of a large number of components where the components are fixed and known, maybe normal, maybe uniform, for example, and the mixture proportions are what we're estimating. So it turns into a maximum likelihood estimation problem. It's now finite, but large number of parameters, and this is a convex optimization problem. And the L's here are just the convolution of the FKs with a normal distribution. So if F is normal, you can convolve that with a normal to get a normal, so you get a closed form for L. If F is uniform, you can convolve that with a normal to get a closed form, well, essentially closed form solution in terms of the CDF of a normal. So you can compute these L's essentially analytically using modern software, and then you can solve this maximum likelihood problem as a convex optimization. So the simplest thing to do is an EM algorithm. It's like 4 lines of code, but it turns out that if you use other modern convex optimization methods, for example, Kunker and Mizera use an interior point method, it can be a lot more work, but a lot faster and a lot more reliable as well in terms of the optimization. And we've been using some sequential quadratic programming methods as well to solve this problem. And so the software implementation is pretty robust. I'd say it kind of, you don't get numerical optimization to a non-global optimum. It's very, for the unimodal case and the scale mixes normal case, I have software here on CRAN. So that's the kind of the background, and so I have to say that when I first came across the normal means problem, it looked to me like a toy problem. It looked like the kind of toy problem that statisticians dream up to study and prove theorems about and teach to undergraduates. And it's only recently that I've become a little obsessed with what I can actually do with this hammer I now have. So I kind of feel like I'm a bit like a kid going around with a new hammer and trying to hit stuff with the normal means problem. But there's a lot you can do with it. And so essentially, every time I see a problem where people are doing some kind of sparsity or shrinkage induction by some kind of thresholding approach or something like that, I'm looking to try and see, can I replace that thresholding approach with this? That's the kind of the, I've devoted several graduate students to like doing this. Okay, so the first application I'll talk about is the multiple testing or false discovery rate. Was there a problem? Question?
Ask whether you have a position for me.
Sure thing, we can talk later. I'm not actually going to talk about the wavelet denoising stuff because that's a little older, and there's some really nice work by Ian Johnston and Bernie Silberman that you can read about, and we have a preprint that does some updates on that exploit the heteroscedastic case. I won't talk about it though. I'll briefly touch on this correlation estimation idea, and then I'll talk about matrix factorization. So in multiple testing, I work a lot in genomics applications. Multiple testing comes up a lot there, so you can characterize what genomicists do is as measuring lots of things with error. They go about, they like doing that, measuring lots of things. There are lots of things, and there's lots of error. So this yields, they typically yield some kind of estimate. I'm going to call it beta hat for something they're interested in, beta. And they usually have, because they've measured multiple things on each beta hat to get their beta hat. So beta hat could have come from a linear regression of something on something, for example. So they often come with their standard errors. So an example would be, if you want to, a simple example would be, they measure the expression or the activity of a bunch of genes, say 10,000 or 20,000 genes, in humans and in chimps, and they want to ask which genes are showing a biggest difference between their activity in humans versus chimps. Okay. Or they may take two human tissues, like a heart and a liver, or multiple hearts, multiple livers, and ask which genes are showing the biggest differences in activity between heart and liver. So then beta is some representation, quantification of the difference in the activity between heart and liver, and they've got an estimate of that and a standard error. So we were looking for the betas that are significantly different from zero, if you like. And so you can see, you can see this is ideally suited, if you like, to the empirical Bayes normal means problem, because we've got these beta hats and their standard errors. And so specifically, the part of the posterior you can compute is the posterior probability that beta is from the non-zero component given the data. So okay, so if there's a non-zero component, we have to include a zero component, right, in order for this not to be trivially 1. So it turns out that there's some identifiability conditions there because you can't, or issues there, because you can't distinguish between something that's exactly 0 and something that's very close to 0. So there are various ways around that. One way is to introduce a small penalty term that encourages mass on the 0 component rather than on the very, very close to 0 component. Another way is to, instead of computing this probability of being non-zero, which is quite delicate and sensitive to the prior on being 0, you can compute something that's less sensitive, like what's the posterior probability it's positive, what's the posterior probability it's negative, and only look at effects where you can be confident in the sign of the effect. And that turns out to be a more robust, that quantity is less sensitive to modeling assumptions you make about, because of this prior, this difficulty of distinguishing between something that's exactly 0 and very close to 0. But, okay, so there's a paper I'll reference later where you can look at more in how we deal with those issues. But just to give you a kind of a sense for, this looks so obvious, I want to tell you what people actually do at the moment in genomics and then just show you how it's different. So this is one common, it's not the only one, but this is one common pipeline people use to do this kind of analysis. They basically compute z-scores from the beta hats and the standard errors, or maybe t-scores if you prefer, turn these into p-values, and then they use this method from John Story to identify findings that are significant at a given false discovery rate, or put another way, to estimate the false discovery rate at any given threshold. So I'll just tell you a little bit about how that method works so that we can contrast it. So here's a distribution of p-values you might get from a genomics experiment. So you can see these are pretty well-behaved p-values. There's lots of them that look kind of uniform, but then there's an excess of them down towards 0. Presumably these are the ones that we're interested in. And so the goal here of q-value is essentially to decompose this distribution of p-values into a null component and an alternative component. So the way it does that is essentially it looks at the right-hand side at the p-values near 1, and it says, well, I'm just going to assume that all the p-values near 1 are null, and that's going to allow me to draw a horizontal line that represents the uniform distribution of p-values, because under the null distribution, under the null hypothesis, the p-values will be uniform. So basically it draws this horizontal line here, and having done that, it says, well, the rest of them must be from the alternative. So now at any given p-value threshold of 0.1, I can estimate my false discovery rate by the area under the dark blue here divided by the total area here. So that's how q-value works. It's kind of a adaptation of the Benjamini-Hochberg procedure. Might not look like it, but it's kind of a generalization to allow for the fact that not all effects are 0, or to exploit the fact that not all effects are 0 to improve power. So I've actually, I really like this procedure because I'm not a big fan of p-values generally, but I find that this kind of gives you a rational way of choosing thresholds for p-values, so I really like this procedure. But I was kind of shocked when I first saw this plot, which is the same plot but put into z-score space. So what I've done is these p-values came from z-scores, so I've just plotted the same plot in z-score space. And so the blue ones are the null. They have a normal distribution. It's not so. I put them on top now, so you can't really see it's normal, but I'm telling you it's normal, just as it was uniform in p-value space. It's normal in normal 0, 1, in fact, in z-score space. And I put on the bottom, I put the alternative distribution, which you can see has this kind of rather strange dip at 0 and bimodal effect. And so what essentially this alternative distribution could only arise, I mean, it's possible that it's not even possible under any scenario, but at best it's only going to be possible if there are no effects less than about 2 or minus 2, right? That all the effects are lying really away from 0. And what we know in genomics, at least, is that as well as many things being close to 0, there are a lot of things, at 0, there are many things close to 0. And the bigger the experiment you get, the more you see, the more significance you get near 0. So what we actually believe is something closer to this, which is what you get if you apply the empirical Bayes normal means approach with a unimodal prior on the effect sizes. So you can see the difference here is that we're explicitly modeling the effect sizes, the effect size distribution, under the alternative. So q-value never really models that. It just takes away a uniform and says what's left is the alternative. So we're making some additional modeling assumptions here. And the modeling assumption we're making is that the effect sizes are unimodal. Sorry, the true effects are unimodal. If you're willing to make that assumption, you get an increase in power. So in this example, this is back in p-value space. Instead of a 12% FDR, for the same data, you get a 5% estimate of the FDR. Of course, we've made additional assumptions. But if you're willing to make those additional assumptions, you're going to get increased power. But there are also some other advantages, I think, of the empirical Bayes normal means approach. One is that we can account for differences in the standard error of different observations more effectively. I didn't do that in this example. I had the same standard error for everything, but that can happen. And on top of that, you don't just get yes-no answers, you get posterior distributions. So you can actually get effect size estimates and credible intervals that take account of the shrinkage of the big effect. So if you're interested in more, I have a biostatistics paper in 2017. Happy to listen to any criticisms or comments or questions at this point before I... Okay, so this one's more in the spirit, this one's a simpler application actually, and it's kind of more in the spirit of replacing a simple thresholding procedure with something that I think might be a little bit better in practice. So it's also a problem that's kind of, it's one of these applied problems that comes up where you're wondering what to do because most of the stuff out there that you're familiar with won't work. So this here is a image plot of the correlation between the gene expression measurements, so gene activity measurements, in different pairs of human tissues. So we've got there are roughly 50, just under 50 human tissues here. Some of them are brain tissues. Actually, all the brains are up here in the top left. And there's heart, liver, lung, and these have been measured on a group of donors, individuals who donated their tissues to science. And for each pair of tissues, we've just computed the correlation across the individuals of, for example, how expressed is this in heart versus liver. Right? So for each pair of tissues, we've got a plot, heart expression versus liver expression. But we only have the data for some individuals won't have given their heart, and other individuals won't have given their liver, or they will have given their heart to someone who needed the heart, and the liver will come to the scientists to do these experiments on. So there's a lot of missing data in the tissue by individual matrix. Okay? Actually, 30% of the data is there and 70% is missing. Okay. So, and some of the tissues have very low sample sizes. So there are some tissues that not many people have donated. Maybe there are only 5 people donated that tissue. So anyway, so that's what we've computed. So each dot here is an estimate of correlation. The number of individuals that are contributing to that correlation estimate is varying from dot to dot. Now, we don't necessarily expect activity to be correlated between your liver and your heart. Maybe it will be for some genes, and maybe it won't be for others. But if there is a correlation, we usually expect it to be positive. So we don't usually expect to see that the individuals with highest activity of a gene in their heart have the lowest activity of that gene in the liver. Biologically, that's not what we expect. So most of the blue points here are probably just noise, sampling error, essentially. So that gives you a sense for how much sampling error there might be. And I'll also note that the blue points tend to preferentially occur in the tissues with a very small sample size. And what we want to do is get a better estimate of the underlying correlation matrix. So the difficulty here is that most methods for shrinking correlation matrices require complete data. You know, they want a sample correlation matrix. We don't have a sample correlation matrix. We just have pairwise the sample correlations. And they also can't take account of the fact that some pairs have because if you have a sample correlation matrix, every observation has the same standard error, right? Whereas here, some points have more standard errors than others. So the idea is we can just turn this into a normal means problem, and the way we can do it is to use the Fisher transform. So the Fisher transform of the correlation is just this. R here is the correlation. So Rij is the population correlation between tissue I and tissue J, and R hat Ij is the observed correlation in the sample correlation between I and J. So we have a Z, which we're trying to estimate, and a Z hat. And Fisher in the 30s, I believe, showed that this approximation, at least under a normal assumption for R, and we're just going to make this approximation anyway. And the standard error here, of course, depends on the sample size. So once we've got this, we just throw it into the empirical Bayes normal means problem. So of course, we're ignoring, in doing so, we're ignoring any structure in the correlation matrix. We're ignoring the fact that it's a correlation matrix and that some of these observations are correlated with one another. But the idea is we think we can do better than just, say, simple thresholding, which would set things to 0 if it's less than some threshold and big otherwise, and we don't know what else to do. So this is what happened. So this was before. This is after. These are the posterior means after translating them back into correlations. So we get a posterior mean for Z back to a correlation. So these are all the brain tissues here. So we can see that there's a lot more correlation between the brain tissues. It's obviously in the original data as well. There's a lot less blue. We could have imposed a constraint that it had to be positive. That would be easy to do. We didn't do that just for the purposes of illustration. And you can also, it's kind of nice that when you look at some of these larger values that haven't been shrunk as much, they tend to be between biologically related tissues. So for example, you can't read it here, but these here are all the artery or several artery tissues. This one here corresponds to 2 skin tissues. So these kind of larger values stick out a little bit more after the shrinkage, and you can go back and look at them. This is just one gene. Here's another gene, and the idea here is just to show, at some genes you don't see such strong correlation in the brains as we saw before, and the shrinkage here is kind of learned maybe that there's less overall correlation here, and you get stronger shrinkage at this gene. So that's kind of the idea that you're adapting the amount of shrinkage to the data. This is another gene where you can see that almost every pair of tissues is correlated. You see much higher sample correlations, and so the shrinkage afterwards is much less strong. So, okay. So it's not going to change the world, but it was. I found it interesting that we had these. We had this problem. We didn't know how to estimate correlations, and this seemed like a nice, quick, and easy, at least, approach to get something to begin with.
All the negative correlations went away. Some of them just looked like they were random dots, so fine. But maybe up on the right, there's more of a region of blue dots, and all the blue dots gone away.
In this one, or the?
This one.
Are you meaning here?
Yeah, so it looks like a coherent region. The scatter of dots, fine, get rid of them.
It's because, they're very small sample sizes. So these are the fallopian tube and the uterus, I believe. There's very small sample sizes. Well, you can see that it doesn't. I actually don't believe these blue here that remain. But you can see these have larger sample sizes, and so it does leave some of the negative ones. As I say, we didn't impose a constraint that it was negative. So this is learning that most things are positive, and I think that's true. So I would bet against these being genuine negative ones. They're the kind of ones where if you were really doing an analysis, you might even remove those sample sizes in advance because you don't really believe that you can get an estimate of the correlation from. But then, of course, you have to decide on a threshold. Thanks for the question. And the last application I wanted to mention is matrix factorization. So by this I mean problems of this form, where I've got a matrix that's n by p, and I'm going to factorize it into k, what you might call factors, that are vectors. Each of these L and Lk and Fk here is each a vector. Lk is a vector of length n, Fk is a vector of length p. So this is a rank k matrix here, and we're essentially representing Y as a rank k matrix plus an error term. So I'll skip that slide and say, so this is a pretty common problem. Probably many of you are familiar with the fact that you can think of PCA as solving a version of this problem where you require the L's and the F's to be orthogonal. There are other versions where you apply a different constraint. So for example, non-negative matrix factorization arises if we constrain the L's and the F's to be non-negative, and that often leads to more interpretable results. Another set of models add an additional constraint that there is a sum to 1 constraint on one or other of the L's or F's. And you can impose other assumptions, but another assumption people have tried and found useful is to assume that these guys are sparse. Right? So that's what I'm going to talk about. And then the question is, of course, well, how sparse should sparse be? And in fact, I'm not going to assume that they're necessarily sparse. I'm going to assume that they might be sparse. And what I'm actually going to assume is that they come from a unimodal distribution. So that unimodal with a mode at 0. So this is what I call empirical Bayes matrix factorization. So we add to this model, I'm going to assume the E's are Gaussian, and the L's, each of the n elements of Lk come from some distribution Gk, L, and each of the elements of Fk come from some distribution Gk, F, and these, I'm going to assume they're unimodal. So, and the idea is that we want to estimate these guys from the data. Ideally, we would do that by maximum likelihood. We can't do that actually, but I'll tell you what we can do in a minute. But maybe the easiest thing is to describe the algorithm actually. So essentially, there's an iterative algorithm for solving this, or approximately fitting this model, that's based on solving the empirical Bayes normal means problem. So to explain that, I'm going to just simplify things and go to the rank 1 case. So y equals LF transpose plus E. L is a vector, comes from GL. F is a vector, its elements come from GF. And so essentially what we do is we iterate between, given F, you can get a normal means problem for L. Essentially, these data here, if you knew F, you would have a series of regressions that gave you the estimates of the regression coefficients L and their standard errors. So you'd have an L hat and a standard error for each. L hat J and the standard error J, Sj. So then you could throw that into your empirical Bayes normal means solver to estimate GL and an L. And then given L, you can estimate GF and F, and you can iterate between them. And this isn't a maximum likelihood estimate, but you can show it's actually equivalent to a variational approximation to this problem, if you know what that is. But if you don't know what that is, it's optimizing an objective function. And it's not guaranteed to find the global optimum, it's a hill climbing algorithm. So this problem is no longer convex, it's biconvex. Each of these problems is convex, but the overall thing is not convex. So that's the intuition. The idea is you can solve this problem iteratively, approximately, provided you can solve the EBNM for G. So actually, I should say the algorithm is kind of modular. That's another thing I'm very keen on these days, is modular things. So as long as you can solve the EBNM problem for G, then you can plug it into this procedure and get a corresponding. So if you want to impose a positivity constraint on the F's and the L's as well, we could do that. This is just an example of applying this to some data. So the data are the same data I told before, but they're based on the same study called the GTEx study, where they measured these gene expression measurements in lots of tissues. So what each factor here represents is one column for each tissue, and the height of the column represents a strength of a genetic effect in that tissue. So what we've got is the matrix that we applied this to is each row is a genetic effect or genetic variant, and each column is a tissue, and the entry of the matrix is how strong an effect does that genetic variant have in that tissue. So this factor here represents the genetic variants that have a strong effect in every tissue. So there are some, in fact many of the genetic variants have a strong effect in every tissue, and they would have a high loading on this factor. This factor here represents the genetic variants that have a very strong effect in brain compared with the other tissues. The brain are the yellow ones here. This one. So I've reordered them slightly compared with before. So these are the brain tissues. This factor here represents genetic variants that have a very strong effect only in blood. So if you have a variant that's only having an effect in blood, it would have a strong loading on this factor and not on the others. Or maybe it has a strong effect everywhere, but an even stronger one in blood. So then it would be loaded on this one and this one. This is a couple of skin tissues here. So these are genetic variants that have an effect in the two skin tissues, and this is the esophageal tissue. So your esophagus has some cells in it that are similar to skin cells, so maybe that's what this is reflecting here. These are some other tissue-specific signals. I like these factors here that have two tissues in, because the ones could be kind of hard to be sure that we're getting something real, but the ones that pick out two tissues I'm really confident in, because the two tissues biologically are related. So in this case, this is the two cerebellar tissues within the brain, and these are two heart tissues. And just to show you how the sparsity helps interpretation, this is a different matrix factorization method that uses a nuclear norm penalty to get a low rank approximation. So it's getting a low rank approximation, but it doesn't have, it's not looking for sparsity. Actually, it does a great job in terms of imputing missing data and finding low rank representations we find, but they're not easy to interpret because it's not looking for a sparse solution in the factor space. So you can see that it doesn't it doesn't yield sparse factors. That's not surprising. It's not trying to, but as a result, it's much harder to interpret. Well, essentially they're not interpretable. I should say this first factor is essentially the same as the first one we found. That's because that's not a sparse factor. It's the first principal component. It's very strong signal, and that goes back to the empirical Bayes approach does not assume that it's sparse. It assumes it's a unimodal distribution, but that can have a long tail, and so it can allow for these kinds of non-sparse effects as well. Okay, so to summarize, I think the empirical Bayes normal means problem is really useful for inducing shrinkage and sparsity in lots of cases, and whenever you want to try inducing shrinkage or sparsity, you might consider using it. And if you want to read more, there's software associated with most of these papers, and they're linkable at my website. Thanks very much.
Question?
We approximate the unimodal density with Gaussian scale mixtures. How many components do you typically use?
Okay, so we have two different approximations. One is unimodal, which uses a mixture of uniforms, and the other is scale mixtures of normals, which uses a mixture of normals.
How many components?
So typically, we don't worry about getting too good an approximation, so we use about 16 components.
Heuristics?
16.
For theory, do you need to grow with n? Or?
I would think that for theory, you would definitely want to grow with n. And you can solve this problem for certainly 1,000 components. I mean, we've done that. We don't do that routinely in our applications. 16 is enough for us in practice. But if you want, the software can do 1,000. So you can use it. It's doable. You can use a lot if you want to prove theorems about them.
All right, so yeah, Peter?
This is a more practical question on the GPEC data.
Yeah.
They have these confounders there, right? Did you adjust for them, or what did you do?
So in this, yeah, no, here for these we did not. Well, okay, so we used the estimates of the genetic effects that were obtained by the consortium, and they adjusted for confounders in computing those estimates of the genetic effects. So I guess the answer is we kind of did. We used their effect sizes that they'd adjusted for those confounders.
I mean, they provide actually a list of confounders. I mean, these. So we looked at this data too, and it's kind of actually whether you adjust or not, it makes a huge difference. But probably you have adjusted it already, and
well, we use their effect sizes.
Results, right?
Yeah.
If
you don't adjust, is that the question? Whether we would get answers?
It was the question whether you did something before just running this matrix factorization or not.
We ran the matrix factorization on estimated genetic effects that were obtained controlling for confounders. Yeah. So, but we didn't do that. They did that. I didn't do it. Yeah.
All right, so since we're running out of time, I propose that we take the other questions offline, and thanks to you again.